Today we are going to, I always have to give them time to start recording in there. Today we're going to discuss, again, the hypothalamic-pituitary gonadal axis. However, in this lecture we move from discussing the GnRH gene itself and the peptide, hormone 1, to discussing the promoter region on the GnRH gene. And we proceed through then to the GnRH receptor and its gene in the promoter region, and so on and so forth. So we have a good understanding of how the neuroendocrine axes work, function physiologically, and at a mid to high level of molecular complexity. And of course our focus now is to utilize the skills and techniques that we're familiar with to evaluate primary literature giving details, those minutiae that of course become the exam questions for the upcoming test 5. So what I wanted to do is switch over to the computer and first go to the web page. So we are on the HPG axis, lecture C. So under the course materials, course content, the lecture C is here. It does not state anywhere here that articles are posted. So under the HPG journals, you'll see that there isn't any article posted here at the time of recording this lecture specific for this portion. It jumps from the alternative splicing that we discussed in the previous lecture to the molecular structure of the human GnRH receptor gene, which is the next lecture. So there's no posted article to read for helping you understand this particular lecture. So this link, the HPG axis lecture C, brings up all of the abstracts that I did use to put together this lecture. So it's not a large number of them, so this is not an extensive and definitely not a comprehensive lecture on this topic. I think we've had enough comprehensive lectures where now we can just have these somewhat choppy lectures where maybe they don't have the best flow that we would like to see from one matter to the next, but at the time putting together these lectures, it was the limits of the information that was available. So the interesting part, of course, would be whether or not the gaps between some of these choppy pieces that we're discussing now have been filled by more recent publications. Again, I'm not so concerned that everything is up to date, because the course is more of a model to understand and appreciate the progression from an introductory textbook that has way outdated references within it through review articles and then up to the primary literature where we are now. So no one's restricted from going to the literature themselves, I guess is the point, if you want to. If you find an interesting article, send me the reference, and I'll try to incorporate it into the course as we go on through the years in the future. So we're now interested in the regulation of the expression of the GnRH gene. You know that there's two genes, GnRH1 and 2, that alternative splicing can occur. Now let's ask the question about the promoter region and how the expression of those genes, or really now we're focusing on the GnRH1 gene that is transcribed to the long form of the message, which contains the exon 2 represented within the message, and it's in the exon 2 that the GnRH peptide sequence is encoded. So that's the one we're thinking about now, other than accepting the fact that the alternative splicing occurs, and gee, it's an odd and interesting thing. Let's now think about how the GnRH1 gene specifically for regulation of the HPG axis is regulated in its expression. And we know that from our prior studies that in both males and females there is a daily rhythm within the HPG axis. And this is much more pronounced in males because there is not a superimposed lunar or monthly type of a cycle, the menstrual cycle, as we know. So there is a more distinctive daily rise and fall in the levels of typical biological rhythm, typically with the testosterone levels being higher in the early morning than they are later in the day. And similarly in females, but the more challenging, let's say, I was going to say interesting, maybe for females, the events in the menstrual cycle aren't more interesting, but anyway, from my perspective it is, because it is a series of events that take place over days rather than thinking of, gee, within 10 minutes or an hour these changes occurred. These are gradual events on the molecular level that change over a matter of days, as we know, building to the LH surge that is responsible for, and obviously comes about from a switch of negative feedback to positive feedback, and then the LH surge is directly responsible for the ovulation and triggering the luteinization of the corpus luteum. So this is a key aspect of reproductive capability in animals that have a menstrual cycle, and similar regulation of LH surge for those in the estrous cycle. Without that, there wouldn't be reproduction. So the male gamete is almost always going to be present and available, as we all know. So much of the attention is focused on the female reproductive cycle. It is more complex and more protracted, more easily targeted with hormonal-based birth control mechanisms. There's advertisements on the television now for a new type, just it looks like a little plastic ring. I can only assume from the advertisements that it is inserted into the vagina and is placed around the cervix where it protrudes into the upper portion of the vagina, and it's filled with hormones, so it's still a hormonal birth control approach, still using basically the same hormones. If you see the advertisement or if you see it in Time magazine or something like that, you take note, you'll see the norestradiol. Is that it? I can't remember those two molecules that are on the exam coming up. But the ones that we've discussed in class, it's still the same, basically the same molecules with that triple bond on them one location there that are being used. Just different ways of getting those hormones into the systemic circulation of human females to prevent the LH surge and prevent ovulation and act as a contraceptive. So obviously I don't need to go on anymore to reinforce the idea and the popularity of focusing on the female reproductive cycle. Also, because it's such a distinctive event that can be induced, as we'll see, then you can study, you can induce it, and then have control over the event, the isolated event, and then you can really get your research tools to focus on what is occurring. There's still a lot we don't know about it yet. How really does the negative feedback turn into positive feedback? We know many events that must take place, like increased sensitivity of the gonadotropes to the GnRH, receptor upregulation, increased expression of the LH alpha and beta gene subunits. And then when you see we go through our discussion, we're going to talk about estrogen acting through ERE to downregulate gene expression. as part of negative feedback. So how does that ultimately get switched to a positive feedback to increase the synthesis of the LH that would be needed so that it could be dumped over the time period that it is, it's a day or so, to produce the LH surge. So there's still a lot that we don't know. Maybe there's more known than when I was last in the literature studying it extensively. But this first discussion that we have really originates from the ERA, the 1997, it's from Brain Research Bulletin, where there was a vast interest in the immediate early genes. And remember, those primarily are C-Phos, the June family, C-June and B-June. Those are a lot of the immediate early genes that received attention at that time. This one particular article is focusing on the GnRH system of seasonal breeders where they can induce the LH surge. So the C-Phos protein actually is what they were looking at, the gene product of the C-Phos gene. It was found to be expressed in nearly half of the GnRH parakarya at the time of the LH surge. But similar to the distribution of the neuroendocrine cells themselves that can be detected by staining for GnRH, the Phos-expressing cells are not limited to a particular anatomical localization. So we've talked about the preoptic area, anterior hypothalamic region, as being a location of GnRH neurons, and the arcuate nucleus. And that's great for purposes of saying there is a location, but they're vastly distributed. We talked about the expression of the GnRH1 gene in the previous lecture, in the olfactory region, the striatum, even in other organs. But the GnRH neurons are somewhat unique in that they're kind of scattered. You find trails of them out through the olfactory regions and things like that. So that's the point that's being made here. You don't have this distinct, like the paraventricular nucleus, the CRH neurons, this distinct butterfly-shaped nucleus, you know, the wings on each side of the third ventricle. So this is the point that's being made here. Similar kind of a distribution of the cells that contain the immediate early gene product, as well as the GnRH neurons. But what is most important is that at the times throughout the estrus cycle, these are seasonal breeders, so they're estrus animals, estrus cycling animals. Other than during the LH surge, you don't find this Phos expression in the GnRH cells. You do find it in cells throughout the preoptic area, anterior hypothalamic region, and the arcuate ventromedial hypothalamic nuclear regions, but not within the GnRH neurons. So some genes are being targeted by these IEGs, but it can't be anything in the GnRH neurons because the protein, the transcription factor proteins are at undetectable levels. So because they may be expressed for only a short time after the initial activation, this research group acutely stimulated the pulsatile LH GnRH secretion with an antagonist to the endogenous opioid peptides. Immediately in your brain, you should be shooting back to that figure in the text that showed the input to the GnRH neurons with the opioid terminal forming an axoaxonic presynaptic synapse, axoaxonic synapse, and through that mediating a presynaptic inhibitory effect. So if you throw in an antagonist to the endogenous opioid peptides, this Win 44441-3, or just Win, it will block the opioid receptors during a phase of the estrus cycle when they should be actively occupied by the opioid being released. I think dynorphin was one of the opioids that we saw in those illustrations. So you get the antagonist in there, blocks the receptor, and it disinhibits the excitatory drive to the GnRH neurons, and you can pharmacologically induce in these animals that are within estrus the LH surge. So this was the approach, during the luteal phase, of the estrus cycle, not during estrus. And you can induce the LH surge. So it's known that in the ewe, the progesterone, and that's the sheep, and I'm not saying in you, I'm saying in ewe. I know you can't appreciate that, but most students in their comprehensive exams, as you're doing in the practice. So this is your LH, this is your ovary. I don't have any ovaries. So I'm not saying you here, I'm saying you. The progesterone inhibits the LH pulse frequency via the EOP neurons, so that the injection of the EOP antagonist directly into the luteal phase produces an immediate increase in the LH and the GnRH pulse frequency. And it should be turned around the other way. You are disinhibiting the GnRH neurons. They take on a higher frequency pulse firing rate, and thereby secrete more GnRH. And that, of course, is not only at a higher frequency do they release GnRH, but at a higher pulse, a higher amount, per pulse. So there's more GnRH getting to their GnRH receptor on the LH gonadotropes, and it's coming more frequently. So with each pulse there's more, and it's coming more frequently, and that is exactly what happens during the portion of the phase, the preovulatory phase, just before the LH surge. So the estrogen's been working on the gonadotropes to build up the GnRH receptors. So when this pulse frequency and concentration increase comes, plenty of receptors to respond to it. And the estrogen has also been acting on the LH alpha and beta genes, so that plenty of LH is being produced and stored in anticipation of this big release event that's going to occur. So they may have generated an LH surge, but it may not have been of the same magnitude because the estrogen might not have been acting at the pituitary gonadotropes. But still, you can induce it at the GnRH neuron, and that's the focus of their attention. And even though they could produce the effect at the GnRH neuron, they still got an increase in the LH. It might not have matched the peak plasma concentration in a normal LH surge, but they mimicked it pharmacologically centrally through blocking the mu-opioid receptor. So this figure is showing some coronal sections through the ewe hypothalamus. You can see the optic tract right here, so that's part of the landmarks for the hypothalamus. And these are double stained, first with an antibody to the CFOS protein. It's a transcription factor, and therefore you expect it to be localized predominantly within the nucleus. So I don't know if you can differentiate all these little dots scattered around on here. That's just nuclei. Certainly at higher magnification in panels B and D, you can see the FOS protein filling the nuclei of cells, but you can't make out any cytoplasm around it. Whereas the GnRH neurons, here in the panels A and B, these are the control ewes that receive just the vehicle injection that's used to deliver the progesterone, excuse me, not the progesterone, the opioid antagonist, the WIN for the animal shown in panels C and D. So you can still see the unstained nucleus of this GnRH neuron in this animal. So This is kind of a typical way of showing this. I would prefer to have seen maybe 3 insets showing 3 separate neurons with GnRH positive cytoplasm and CFOS negative nuclei. But, you know, 1 is okay, I guess. And then here in panel C and D, we see similar levels of the hypothalamus with staining, again for the GnRH and for the CFOS protein. And they're picking this one particular GnRH neuron here and showing the cytoplasm stained, and you cannot see the empty spot where the nucleus would be because it is unstained, so it is CFOS positive. So it's only during the portion of the induced LH surge that the CFOS immediate early gene protein is detected within the nuclei of the GnRH neurons. And as stated in the previous slide, it was about half of them that contained it. So the question I have for you to answer in your own mind is, if the CFOS is there, and we're suspecting that the CFOS is responsible, remember we have disinhibited, so we're increasing the frequency of action potentials per pulse and the number of pulses of action potentials in these neurons. So that tells us release. We've increased release, so we must increase synthesis. So first concept put forth by me in this course for neuroendocrinology is synthesis and release, or release and synthesis. They're coupled. They're always coupled. So when we think of the synthesis for the peptide hormones, we got to get to the gene. So the immediate early genes are appearing in these cells when we know there's a tremendous amount of release of GnRH, and we see the CFOS there. So then we should be predicting that there is a consensus sequence on the promoter region of the GnRH gene. Do you remember which one by acronym is the regulatory element or the sequence of DNA that the CFOS binds to? AP1, very good. Very good, everybody remembers. Okay, so that one brings us, so it's, when we get to the pituitary, it's always going to be the CRE, because every hypothalamic releasing hormone receptor on its pituitary trope target cell is coupled at least to adenylate cyclase, and increases cyclic AMP, increases phosphorylation of Krebs ultimately, and their promoter region is going to have a CRE on it, 1 or more CREs. That's a given to link release and synthesis at the level of the anterior pituitary. But here, looking at gland 1, neuron 1, if you will, GnRH neuron, it may be an AP1 at least for coupling the modified input during the LH surge. The estrogen receptors then bring us initially to think that we're talking about negative feedback, and that indeed is the idea. So we know when we read the descriptions that are at a graduate level of sophistication, let's say, describing the role of estrogen in the menstrual cycle, that a positive feedback is shown, and there's little mention of its role in negative feedback, because progesterone plays such a potent role in negative feedback, especially, of course, in the luteal phase, and especially for LH. And why would I say especially for LH? Is there a particular hormone 3 that targets specifically FSH for negative feedback? Inhibin, very good. So there is a separate negative feedback system from the gonads to the anterior pituitary, selectively targeting the FSH. So when we talk about negative feedback of the steroids at the level of the anterior pituitary, it's really for the LH. And that's why you see, we will see at that point the estrogen receptors there. So we think of the estrogen mediating the negative feedback in general, but somehow at the level of the anterior pituitary anyway, for the LH, it can change from negative to positive. The level of the GnRH neurons, the expectation is that it's a negative feedback. And the evidence as it emerged over the years, you remember the initial method we talked about for visualizing cells in the brain that contained steroid hormone receptors was to inject the animal. You'd either remove the endogenous source of the, and it was the most likely approach, the endogenous source of the steroid, and inject the animals with, once those levels decrease, then inject the animals with radio-labeled hormone, give some time for it to get into the brain and bind to its receptor and be concentrated in neurons. Then you take the brain tissue, section it, expose it to emulsion film, develop that, and then you can visualize where in the brain you have these steroid hormone concentrating neurons. And it was just assumed that, actually originally they weren't thinking of receptors, but then ultimately these are the receptor containing cells, but it wasn't until the receptor proteins and their genes were discovered that we could identify specifically and molecularly through a variety of techniques. We'll see one with immunocytochemistry and in situ hybridization, whether or not the estrogen receptors or the steroid receptors were expressed, genes were expressed in those cells that concentrated the steroid hormone. So kind of historically, that's the way it went. With utilizing all those methods being kind of thrown at the reproductive system, again, controlling reproduction in humans is extremely important around the world. Population growth is a tremendous concern in every nation, and it's an individual concern to be able to make individual decisions. So a lot of research has gone on in this area. So there's a tremendous effort to understand the entire system. So from those approaches, it was determined or hypothesized that the estrogen receptor gene is not expressed in the GnRH neurons. And most recently, and in this particular study, the vast majority of the GnRH neurons in the sheep, as in other mammals, do not contain the estrogen receptors. So when you hear this sequence of words, this phraseology, do not contain estrogen receptors, then you know they're looking for the presence of the protein. Otherwise, it would say do not express the gene for estrogen receptors. So if you're looking at message, then you would say, you use the term gene expression, not anything about containing the estrogen receptor. The estrogen receptor is a protein, so you have to be looking at that protein and find it contained within the cells to say they contain estrogen receptors. But if you look at the message, then you're saying they express the gene for the estrogen receptor. So we would expect then that these investigators were using an antibody to the protein for the estrogen receptor to visualize it. And indeed, that is the case. Here's the article: gonadotropin-releasing hormone neurons in the rhesus macaque are not immunoreactive for the estrogen receptor. So we've obviously switched papers. The previous paper, they also demonstrated that the immunoreactivity for the estrogen receptor is not within the GnRH neurons, within the ewe, as in other mammals. Here is a subhuman. Is the macaque a primate? I don't think it is a primate, is it? A monkey, anyway. It is subhuman, but not a subhuman primate. They're also using immunocytochemical approaches, staining for GnRH and for the estrogen receptor could not find them co-localized. So, therefore, the negative feedback influence of estradiol during anestrus is likely conveyed to GnRH neurons by afferents. So there's some way of triggering the neuronal systems that, or targeting, the neuronal systems that dump neurotransmitters onto the GnRH neurons that allows the negative feedback to be conveyed to the GnRH neurons at that level of hormone 1, gland 1. So this is one way of feeling a little bit more comfortable about the possibility of a switch from a negative to a positive feedback at the level of GnRH. You're not trying to figure out, well, how can estrogen act on the GnRH neuron, the receptor within there, and suddenly change it from a negative to a positive feedback. You have to think that way at the level of the gonadotrope and the pituitary. There's something interesting going on there where there is a real switch. But with the GnRH neuron, you've just got differential neuronal circuitry where you might have estrogen receptors of different affinities. High affinity at low concentrations, at low numbers of receptors, concentration of receptors, and low affinity receptors at higher levels, higher concentrations or numbers of those receptors. So when you have low levels of estrogen, the high affinity receptors are producing negative feedback maybe, and then when estrogen levels get higher, they occupy more of the low affinity receptors, and then those contribute to the positive feedback, or differentially distributed in different neuronal circuits. So the low affinity receptors in the neurons that are the drive to the GnRH neurons, the excitatory gas pedal circuits. So when estrogen levels get high, acting at those receptors to increase the gas pedal. But at low levels, it's not affecting those, it's only affecting the opioids that would cause the inhibition. So it's easier to come up with ideas on how you might have these differences. Of course, at this time, it was only thought that there was one estrogen receptor. The realization that there's at least two different estrogen receptors hadn't emerged in the scientific community yet. So the distribution of the estrogen receptor containing cells that project to the ovine median eminence are shown in this figure. So this is a classic approach. A lot of cells in the hypothalamus. It's an endocrine tissue. It's not an organ. It has a mixed function, and only those hypophysiotropic neurons regulating the anterior pituitary will project to the median eminence, so you must identify those first and then look at their chemical nature and then say, okay, these are hypophysiotrophic neurons controlling the anterior pituitary, and this is their chemical nature. They either have the estrogen receptor in it or they don't. They either have the peptide, the releasing hormone in it, or they don't, and things like that. Well, one way to identify those hypophysiotrophic neurons in the hypothalamus is to inject a tracer, a neuronal tracer, into the median eminence that will be picked up by the neurohemal terminals and transported back to the cell body, called retrograde transport, track tracing. Visualize that tracer and then look for the other chemical markers within those cells. It's not easy injecting the median eminence. Really, really difficult to get to it. It's at the base of the brain, right in the center of the skull, even for sheep. Tremendous amount of effort to get this little injection in there. So it's always highly regarded level of work, technical sophistication. Individuals that used to do this a lot were, appreciated by everyone that never wanted to have to try to do that. So those types of things. Anyway, it is the standard test to be able to say this particular neuron that contains this cholera toxin immunoreactivity is hypophysiotrophic because it projects to the median eminence. Then if it is also stained for the estrogen receptor, then you can say this is a neuron, the hypophysiotrophic neuron projects to the median eminence, and it contains the estrogen receptor. Or you can take that theoretical jump and say it expresses the gene for the estrogen receptor. Then it would be very important to try to show if any of those with a third stain were also to contain the GnRH. But this one at least shows that there are estrogen receptor containing neurons in the hypothalamus that project to the median eminence, and therefore there's a potential for, and as well, of course, they're located in the medial preoptic area and the ventromedial hypothalamic arcuate nucleus area, where we know the GnRH neurons are also located. So there's a potential for that estrogen receptor to be within a GnRH neuron that also projects to the median eminence. So that establishes that. In that study, they were not able to have a triple label, which is easier to do today with all the available different fluorescent markers and confocal microscopy. You could produce beautiful pictures, but you might have a difficult time convincing the IACUC why you'd want to do this on sheep to utilize that animal. Anyway, let's move on. So the gonadal-steroid receptors and the regulation of GnRH secretion in farm animals is the source of the information shown here. So I think you're seeing this agricultural significance of this, tremendous impact on the potential for producing more and more agricultural or economical domesticated animals. And this emerged at the time that the alpha and the beta types of the estrogen receptors were known. So therefore they could generate a cDNA probe. So they not only knew the gene sequence, they knew the messenger RNA for both of these isoforms of the receptor, or subtypes of the receptors, I should say. And of course the protein sequences. So at least you could use in situ hybridization to generate your probe to differentiate between the two of them. I don't know if there are separate antibodies that are available now. I don't know what the real differences are in the proteins themselves, whether they differ significantly enough where it'd be easy to generate antibodies to them that would be specific and not cross-react with the other one. So these would be the things that we would be interested in. But you easily can do that with the message, coming up with a cDNA that would be specific for the distinctly different parts of the messenger RNA for these two receptors. And what we're seeing here, of course, is that in both the ewe and the ram, Ba-ram-ewe. Anybody see that movie? That'll be enough, pig. Anyway, the estrogen receptor alpha seems to be in both gender, the male and the female sheep, the predominant one in the hypothalamus, whereas the estrogen receptor beta is less common in those animals. They went on with this method to counterstain with, in a table that reviews the localization of the estrogen receptors, referring to those articles where the researchers did do the double staining of the estrogen receptor or double identification of the estrogen receptor and potential transmitter marker in those cells. They're able to identify the estrogen receptor in neurons that contain these neurotransmitters or markers for a particular type of transmitter within these nuclei, and determined a certain percentage. As I said, these are the references for the actual work. So let's take a look at the list. The tyrosine hydroxylase for dopamine, that would be the arcuate nucleus, which is what we're seeing here. A lot of these estrogen receptors very likely are in dopamine neurons and not in GnRH neurons. Preoptic area, of course, there are not any tyrosine hydroxylase containing neurons there, so you'd have a 0. There's no dopamine neurons there. The anterior hypothalamus, where you do have more, there's a certain percentage there. I guess in the preoptic area there are some dopamine neurons called the A14 region. That wasn't even on our list when we were doing that introductory discussion there. One group saw 13 to 18%. Anyway, we don't need to focus so much on those details. GABA in the preoptic area, they stained with an antibody against the synthetic enzyme, the GAD. Remember that from neurochemistry, glutamic acid decarboxylase. So in the preoptic area, about 44% of those. So estrogen receptor in one of the primary inhibitory type of neurons using GABA as a neurotransmitter. So we'd predict that estrogen would increase the expression of the GAD genes. There's 2 different GAD genes. And we'd predict that there's an ERE on the promoter region for GAD, and that would be an activator, not a repressor. Beta endorphin neurons in the arcuate nucleus, 15 to 20%. So that's a kind of a low number, might not be involved in the regulating the negative feedback. Neuropeptide Y also in the arcuate nucleus, only 3%. Somatostatin, about 70% of the estrogen immunoreactive cells. So I don't know what that means relative to the HPG axis. Dopamine beta hydroxylase staining indicating noradrenaline or norepinephrine, as we know we are now supposed to use. In the ventrolateral medulla, the A1, and the nucleus solitary tract, the A2 levels, a range of percentages, 20 to 70, 4 to 20, 16 to 60, and 8 to 25. So a lot of double labeling of the estrogen receptor with the marker for the norepinephrine synthesizing neurons. And then in the GnRH neurons in the preoptic area, 0%. So this table reinforces the idea that estrogen, if it's involved in negative feedback at the level of gland 1, hormone 1, is mediated through indirect mechanisms, the neuroendocrine transduction mechanisms that we had discussed.
I've got a question.
Yes?
What if they look at membrane level? They just look at the nucleus, right?
Membrane level of what?
Receptors at the membrane. Because they...
No. Remember, this is a steroid. The question was whether they looked at membrane receptors as well as cytoplasmic or nuclear. And of course, the steroid receptors are either cytoplasmic or nuclear. And the estrogen receptor, upon synthesis, is translocated to the nucleus, so it's only a nuclear localization.
There are some research that look at that stuff.
Binding sites. Now, there's a difference between binding proteins that bind estrogen in the membranes and receptors that are truly identified, right? So the proteins or the molecules in the membranes that can bind the steroid hormones and produce more acute effects haven't come to the point where we know exactly what they are. They're not isolated and purified and sequenced and completely characterized all the way down to the level of the genes that control them. So we don't call them receptors yet, and we don't know what mechanism or coupling complex is involved to bring about the functional response. So until we know all of that, we don't call those estrogen binding molecules in the membrane receptors. They're binding sites, but they're not receptors. Okay? So at this level of it, in this era of the investigation, we're just discovering the RNA sequences, the mRNA sequences for the 2 estrogen receptors. In situ hybridization was available, northern blot. So a lot of the more contemporary techniques of real-time PCR wasn't available, wasn't popular at these times. So you really couldn't quantitate. But the biggest concern was the anatomical localization. They didn't care if they were membrane or nuclear or cytoplasmic. Are they in the GnRH neurons? And they weren't. They were in other neurons. But what we want to see is, as we proceed, we'll find that the promoter for GnRH actually has ERE, I think more than one ERE on it. So if they don't express the gene for the estrogen receptor and contain the estrogen receptor, then why would the gene have the ERE on it, right? And it is functional and everything. So I think the present methods or the particular time in which the estrogen receptor gene is expressed just had not been discovered at the time I looked in the literature here. So I don't know if anyone has found the estrogen receptor gene being expressed in GnRH neurons since then. But when it's difficult to find, then you know that it's performing a very specialized function. So the progesterone receptors in the GnRH neurons, so of course progesterone is definitely a hormone 3 that acts through negative feedback at the level of gland 1, hormone 1. So there's negative feedback actions that are mediated by the classical nuclear progesterone receptor, and the neurochemical identity of the PR containing cells in the brain of the domestic animals is unknown. So that was their justification at the time of publishing this. This is a film autoradiogram of the progesterone receptor mRNA, so in situ hybridization of the message. So we see that the progesterone receptor gene is expressed in both the RAM and the EU in the preoptic area and in the arcuate and ventromedial hypothalamic areas, where in this species, anyway, we know that the GnRH neurons are localized. So this study had not done the co-localization study, just showed, remember at this time, in situ hybridization was a hot method. We could ask the question, what neurons in the brain express this particular gene? So this study established that part at least. So I don't know if since then the progesterone receptor gene expression and its protein, the receptor itself, have been found to be co-localized in the GnRH neurons, if it's particular for any time during the day in both males and females, and if it is particular for any phase of the menstrual or estrous cycle. Well, let's start thinking a little bit about the GnRH promoter region. We're now at the point we have the skill when we start thinking about things and read about events that occur. We can predict what we would expect to find on the promoter region. So the classical approaches were used, and 2 activators and 2 repressors were found, both a proximal and a distal activator and repressor area was found. So we can now predict how these were evaluated. Probably transient transfection with the truncation method that we're talking about. We get activation until we clip to this point. We get repression until we clip to this point, and so on and so forth. So they seem to alternate. The distal repressor way out 1.5 kilobases. The distal activator about a third of that distance. And the proximal repressors, minus 73 to 16, actually more proximal, closer to 0, remember, than the proximal activators. The little bit different in the rat, but that's not of great concern for us. Just always reminding us that there can be species differences. The 426 base pair long palindromic sequence can form a cruciform structure. So this is reminding us that the sequences in the promoter region allow for actual conformational changes of the promoter region, bringing repressors in or bringing activators together to increase the likelihood or decrease the likelihood of the transcription complex getting to the start site. So then the basal promoter activity of serial deletion constructs of the rat GnRH promoter is measured by luciferase activity in GT1. Now, this is a neuronal cell line, behave like GnRH neurons. So using a reporter gene, we're familiar with that approach now. So here you see 3002 length, base pair length for the promoter region all the way down to 81. And we just follow the percent of the largest construct. So the largest was set at 100% relative luciferase activity. Make it a little bit shorter, and you actually exceed that. You go up to 153%. So maybe a repressor somewhere between these two portions. And you can see that this progressively just drops off to 0% as you approach the 0 point on this line scale. So this is actual data that shows how the percent expression of your reporter gene drops off significantly as you truncate. So there's something important in between these to sustain basal expression. So serial deletion constructs in the rat GnRH promoter fused to the luciferase reporter were introduced into GT1 cells by a modified calcium phosphate co-precipitation procedure that we discussed back with methods, and the data are expressed as means plus or minus standard error of the mean of the percentage of the largest construct. Now this table is showing us a little bit more information about the compilation of the transcription factors and the hormones or second messengers that regulate hypothalamic GnRH promoter in the GT1 cells. So these people did quite a bit of work to map where they are in human, rat, mouse, and to give us a feeling for the transcriptional effect. We don't recognize too many of these up here other than the FOS and the Jun, and we see that it's present on human. So the U is not on here, so we don't know. That's that one study that we looked at. So maybe the U is like the human and does have a FOS Jun or AP1 site on the promoter region. And we see that in the GN11 cells, at least, this is positive. So that's what we predicted, wasn't it? In the LH surge, the FOS expression increases in the GnRH neurons of the U anyway. We predict maybe even humans. And this is an activator or increases, has a positive effect on gene expression. And then hormone second messengers collectively, human chorionic gonadotropin receptor present on the rat, estrogen, the ERE on rat, progesterone on rat. Either these are not known or haven't been detected at the time of this publication on human. And those are the only ones that we really need to bother with. And they are considered to be negative as far as their transcriptional effect. The reference for this, the source, is provided here. And then finally, talk about the GnRH promoter region as far as having EREs. So there's two EREs. They're fully palindromic, 8 to 9 intervening nucleotides between the palindromic sequences, 1.5 kilobase pairs upstream, so this is probably that distal activator, separated by 49 base pairs, so they're pretty close to each other. So how to determine the activation or repression is the question. Well, you transfect human placental genes, cells, with the 1.86, you got to get past that 1.5, upstream of the GnRH gene, and use a luciferase reporter gene. You get an insignificant response to the change in estrogen in the medium. Why might that be? Well, it's because those JEG cells don't express the estrogen receptor gene, so you have to co-transfect those cells with the estrogen receptor cDNA, get the estrogen receptor in there. So this is a convenient cell line for testing estrogen-dependent effects, estrogen receptor-dependent effects. So then after co-transfection, so you've got both of these being expressed in this transfected cell line, then you get a dose-dependent increase in the luciferase, retained in the constructs way out to the 548 base pair, upstream to the transcriptional start site. So as you put more construct in, you get a dose-dependent increase. These are dose-dependent decreases. Sorry. And the dose they're referring to is the concentration of the estrogen in the medium. So it is a repressor. You get a reversed response to estrogen when the 5' deletion reaches down to the minus 169 base pairs. So you get change from a decrease to an increase in the luciferase activity. So this shows us that there might be some way in switching from negative to positive feedback. There'd have to be other proteins that would cover the repressor part, the repressor ERE out here near the 548, and somehow expose the activator ERE to get the switch from negative to positive feedback. But again, this is the promoter for GnRH. So why does it have EREs if the GnRH neurons don't express the estrogen receptor? So I'm not going to worry about this. This is just the approach they use to sequence those estrogen response portions on the promoter region that they were studying to verify that they were the palindromic sequences. You get the ligated estrogen receptor homodimer bound. When you digest the DNA that you've isolated with DNase, the proteins that protect it, and then you can dissociate them later and digest the DNA that you get from that and sequence it, and you know what it was. So they ended up with the palindromic sequences that were mentioned. So that's great. That's in JEG cells that are co-transfected. What happens in neural tissue? So there's CNS tumor cell lines that was produced in transgenic mouse. So you can do the in vitro transient transfection study with little more than 1,000 base pairs in the promoter region. They identify estrogen response element as well. Use that then with the simian virus 40 large T antigen gene to produce transgenic mouse and make a tumor cell line. This is how they produce that GN1 cell line that we saw earlier. So basically they're trying to induce a neuronal tumor in the brains of mice by introducing a tumorigenic gene, if you will, and then isolate the GnRH neurons from the tumors produced in the mouse, transgenic mouse, and those will become immortal cells to work with, so you can get a lot of work done, and you're actually looking at neurons. So also with in vivo studies from those transgenic mice, they put in the 5,000 or the 484 base pair 5' upstream with the luciferase reporter gene. So this is what we were talking about earlier. You can either use this approach to produce kind of a tumor to immortalize cells either without putting in a gene of interest or with putting in a gene of interest. And in this case, it's combined with putting in the promoter region with the reporter gene, and then you've got a transgenic animal that has the promoter region to study, even using the same approach, only 2 different lengths. Harder to produce transgenic animals than transiently transfected cells, and a reporter gene. So there was a positive effect on luciferase with the 5,000 base pair promoter, but not with the 484. So we'd have to go back and look at the location. I think the 484 fell below the one mentioned up here at the 548. So it was shorter. So they somehow lost the positive expression in the cells. It might have been due to estrogen. Might be way out here somewhere farther out, more distally. You have the tissue and cell-specific gene expression. The luciferase does increase with gonadectomy, indicating that removal of extirpation of the gland 3 would decrease hormone 3, and therefore, predictably, remove that from your negative feedback picture, and you should get an increased expression of the reporter gene, because estrogen is not present to bind to the estrogen receptor to bind to the promoter region in the transgenic animals to keep the reporter gene levels low. So 20 to 30% in gonadectomized males, 65% increase in gonadectomized females. So this would be telling us that the ERE on the GnRH gene does respond in a predictable fashion within a negative feedback model in the transgenic animals. So if you replace the hormone, it brings the expression of the luciferase reporter gene back down at least 50%, closer to baseline. So it is the ERE on the GnRH promoter is behaving very much like a repressor, a negative feedback type of a model. So I'll stop there, and oops, there's the students that were here on this day that recorded this. I'm trying to push TJ. Provide you with a sufficient amount of information. Again, when you reach the point where you don't have the full story, you can't put together a complete picture of the story. And at the time of putting this together, that was the state of the knowledge that we had to understand how this system works. So it also lets us work with that ability that we've developed to be able to say, I predict this would be present on the promoter region and that it would respond functionally in this direction or that direction. Appreciate your time and attention, and I'll see you again soon.